This screencast is going to tell you a bit more about photosynthesis, and to do that we are going to look at a cross-section of a leaf. So we know that photosynthesis is important because it's going to fuel ecosystems and also replenish the atmosphere of the earth with oxygen. So we'll talk about this more later on, but the rate of photosynthesis, we can measure that by either the disappearance of a substrate or an accumulation of products. So from the worksheet you did in class, you know that for photosynthesis to happen, we need three things: carbon dioxide, water, and then of course light. And with those, we're able to produce glucose, oxygen, and sometimes a little water as well. Now some of you are probably realizing this equation isn't balanced, so let me just add in our prefixes here, going back to chemistry. So I've just added those 6's in red to make that equation balanced for you. So to measure the rate of photosynthesis, we could either look at the production of oxygen, which we know is released when photosynthesis happens, or you could look at the consumption of carbon dioxide, since that is what is going into this equation. So let's look at the anatomy of a leaf, and do that looking at a leaf cross-section. So a leaf is going to be made up of layers of cells. So let's put some labels on here first. The outermost surface of the leaf is going to have a thin, waxy coating on it, and that here is our cuticle. So that's this lighter green layer that we have in the upper portion of the leaf. The main function of the cuticle is going to be to prevent water loss from within the leaf. Plants that leave entirely within water, they don't have a cuticle. Directly underneath the cuticle layer, we have our epidermis. And the epidermis can be separated in, we either will have an upper epidermis or a lower epidermis, as you can see shown here and here. There's some structures I haven't labeled, and that's sort of in this vein that we have shown here. Keep in mind that this spongy mesophyll that I'm talking about is this whole layer here. Okay, so within that layer we have this vein, this purple structure, and I want to show you what those different parts are. The purple sides are, the purple cells that are lining that vein are what are called the bundle sheath cells, and they're the outermost layer of the vein. They will create a circle around two structures called the xylem and the phloem. And this leaf cross-section doesn't do a good job of differentiating between xylem and phloem, but if we zoom in a bit, usually the xylem is going to be shaded a little lighter in diagrams, and the xylem is important because it's transporting water. Then the phloem will be what is transporting sugars, the food for the actual plant. So zooming back out now, I want to focus on the layers called the mesophyll. So we have the palisade mesophyll here, and then beneath it we have our spongy mesophyll, right? So keep in mind that our vein is running through that spongy mesophyll, but the spongy mesophyll itself has additional cells shown in green there. So mesophyll in Greek means middle, and leaf is also known as phylum. So meso means middle, leaf means phylum. Sorry, messed that up. Meso means middle, phylum means leaf. So we can divide our mesophyll into two layers, as I said, the palisade and the spongy layers. The palisade cells are going to look more columnar, and they're going to lie just under that epidermis. Then our spongy cells are going to be more loosely packed together. As you can see, there's some space in between them. And that spongy layer will lie between the palisade layer and also the lower epidermis. We see air spaces in the spongy mesophyll, and that's going to allow for gas exchange, which will be really important here. The mesophyll cells, both the palisade and the spongy mesophyll, are going to be packed with the chloroplast, which we know is really important because this is where photosynthesis is actually occurring. Keep in mind that we also have an epidermis that is lining the lower area here, right, our lower epidermis. And you also will have a cuticle there, which is not shown. But the leaf will also have tiny holes in the epidermis, which are called stomata. So we see those holes in between those two cells, those are the stomata. And then surrounding those actual holes, we have specialized cells which are called guard cells that will surround the stomata. They're sort of shaped like two cupped hands. So you can see one guard cell that I have there. You also have another one on the other side of the hole. Here's another guard cell and another guard cell with a stomata in between where the gas exchange can happen. So then when there's changes within water pressure, it will cause the stoma, that's the singular of stomata, to either open or close. And if the guard cells are full of water, they're going to swell up and they'll bend away from each other, which will open the stoma. Then when it's a dry time, the guard cell will close. It won't want water to come in or out. So that's all I have to tell you about the leaf for now. I'll put a couple of questions on the next slide for you to sort of help with your comprehension, to see if you guys totally got everything that I talked about. And if you're able to answer all those questions, then you are good to go. So first question, what two tissues are found within a vein? So remember, you're looking within that spongy mesophyll at the vein. There are two different tissues. You should know what mesophyll means, where you can find it. You should also know where chloroplasts are located. Know what is the outermost layer of cells. And know that that is different from the waxy covering that we have on a leaf. So know what the name of that is as well. Also know what are the names of the cells that function to open and close the stomata. Singular is stoma, if you have more than one, stomata. Also know what is the outer layer of the vein called. Know what is the name of the cells that are shaped like columns that you'll find under the epidermis. Know the openings that will allow for gas exchange. And then lastly, know the stalk that connects the leaf to the stem. Actually, no, scratch that last question. Just know the ones that I have listed here. If you have any questions, shoot me an email.